The entire blueprint of our body is encoded in the 4-letter alphabet of our DNA. A lot of valuable information is stored there, such as the fact that you have a beating heart, an incredible immune system, and are able to understand this video with that brain of yours. All of that is stored in stretches of DNA, and there is a lot of it. If we stretch all the DNA of one cell out, it would be more than 2 meters long. Luckily for us, this information is neatly stored in the nucleus of the cell in an organized manner, so that each and every cell can make sense of it all. But how does this organization look like? Well, let's take a look at the nucleus of a cell. Most of the times, DNA is partially unwound as strings of spaghetti. It is a bit difficult to discern these strings. So, let's have a look when the cell divides and the DNA is replicated and neatly packaged into structures called chromosomes. We have 46 of them. If we arrange these chromosomes on size and their banding pattern, you will notice that there are 23 different chromosomes, and that these form pairs of homologous chromosomes, one inherited from each parent. 22 pairs are the same for males and females. Those are also known as the autosomes. The final pair are known as sex chromosomes, as they participate in sex determination and are thus different for males and females. Mind you, the structure of the chromosome is only visible when the DNA is duplicated. These two parts are actually the same and are called sister chromatids. They will be separated after cell division. Now let's return to the unwound state of a chromosome. Each chromosome contains different genes. A gene is a functional unit of heredity that encodes for a gene product, such as a specific protein. As you have 2 pairs of the same chromosome, each gene is present in twofold. We have around 20,000 different protein-coding genes, which is only a part of our entire genome. The regions between the genes are called intergenic regions, and are made up of non-coding sequences. The function of these sequences are not understood very well. Each gene consists of different elements. A promoter, the region that precedes the gene, enables the transcription of the gene. The gene body, or open reading frame, consisting of introns that will be removed and exons that contain the coding sequence for the gene product. On both ends of the gene body, there are untranslated regions, better known as UTRs, that have regulatory functions. All these parts are important for a functional gene. The different elements can be distinguished from one another by the sequence of the DNA. So how does this look? Zooming in on the DNA, we actually see 2 strands that coil around each other to form a double helix. Each strand consists of nucleotides that form a polynucleotide chain together. We can distinguish 4 types of nucleotides that each have a different base: cytosine, C, guanine, G, adenosine, A, or thymine, T. All nucleotides also contain a sugar and a phosphate group that attaches one nucleotide to the next through a covalent bond between the groups. The 2 polynucleotide chains are bound together through the interaction of 2 nucleotides, or a base pair. The C binds to a G and the A binds to the T. Therefore, DNA always consists of 2 complementary strands. DNA can both be read from the forward strand or reverse strand, and the sequence of nucleotides dictates which information is contained. So, our genome is made up of different building blocks, that are all encoded in the nucleotide sequence of our DNA. Together, this holds all information to create a human being with all its complex features.